Welcome, everybody. My name is Aaron Sato. I'm the CSO of Twist Bioscience, and today I'm going to give you an overview of a brand new talk that we've been working on, focusing on how we use machine learning and NGS analysis to really get the most out of our library panning work that we do. And when I started thinking about machine learning a couple years ago, I thought it was a lot of hype. I thought you could get most things through traditional screening. I didn't really see a need for it, but I wanted to give it a shot. So we spent a lot of time on it, we dug into it, and now I'm a believer. I actually do believe now that sequencing coupled with all the things I'll talk about today really help us come up with more leads against the targets that we're going after. And before we actually did all these things, we actually left a lot of sequences on the table that we would have missed through traditional screening. So I am definitely a believer now. And I hope you are a believer too at the end of this talk. So Twist Biopharma, we're a division of Twist Bioscience. We know, and we're really focused and dedicated to our customers, that we know that the race is on and that time is money in many scenarios to drive and find the best in-class therapeutics against all the targets that our customers are focused on. And as you'll see today, by coupling in machine learning tools and sequencing, we really firmly believe that we can help our customers find those key candidates that, again, we would have missed through traditional screening. And again, we're using the same approaches for our optimization work that I'll hint on as well. So as I mentioned, Twist Biopharma, we're a division of Twist Bioscience. You may be very familiar with Twist in terms of a lot of the DNA products that we have, and I'll show you our core DNA technology in a second. But Twist Biopharma basically builds on all those DNA products to create a whole pipeline for using those products to help us discover and optimize antibody drugs. Another division of the company is actually our data storage division, where again, it's an application of DNA, where we're actually using DNA to store data for long-term data storage. So we're continuing to evolve as an organization to use DNA in all kinds of different applications, and actually in our case, apply it as a service that we use to help our customers discover antibodies. So I always say the best companies out there really understand the one thing they're really good at. And for Twist, that's our ability to print DNA. So we have a silicon chip where we can actually print up to 1 million individual oligos up to 300 base pairs in length. And then we can elute off those oligos in pools of discrete sequences, anywhere from, say, 10 oligos in a pool all the way up to 1 million. And we use those pools of oligos to make those foundational DNA products I talked about before. So gene fragments, clonal genes, oligo pools. It's also key to our NGS business, where we use biotinylated oligos to pull down specific gene sets ahead of doing next generation sequencing. So again, it's key and central to all of those technologies. But again, for biopharma, we basically utilize both clonal genes to make antibodies, as well as our ability to use oligo pools to generate libraries to really create our business. And we really sit at the forefront of the discovery and optimization workflow going towards the clinic. As you'll see in a second, we have a whole panel of different fully human antibody libraries in phage display. Now we have up to 18, getting close to 20 that we'll be releasing very soon. And as well as we have a whole platform for antibody optimization that we call TAO, which stands for Twist Antibody Optimization. So if you have any needs in the space of discovering antibodies for your particular application or optimizing an existing candidate you already have, Twist Biopharma can help you with that. When I first came to Twist, I wasn't really sold on the idea of using oligo pools for generating libraries. I didn't really see how much more impactful is it from just using standard, say, trim-based oligos or degenerate oligos. But after a while, I really became sold on this idea that using oligo pools to generate libraries gives you a lot tighter control over the construction of your library. As an antibody and protein engineer, it's often really difficult to think up of a library in your head and then actually work with DNA chemists to actually create that library. But because we are making discrete sequences within oligo pools, we have very tight control over the codons and amino acids that go into our library. And shown here is just some examples of why I believe strongly that using algal pools for generating synthetic libraries is much more impactful over other approaches. First of all, we have very tight base-by-base precision over all the varied regions that we incorporate into a library. Of course, since we're using discrete codon sequences, we have tight control over the codons as well. One huge advantage that I see in terms of building antibody libraries is we have a lot of control over the length of the CDR loops that we're varying in the context of a single human germline framework. We can, of course, avoid restriction sites as we build the libraries, precisely control the combinatorial diversity of the genes and sequences that we're shuffling, as well as the ratios of amino acids in each particular position of the library. Of course, we can use multiple germline scaffolds if we're talking about antibody libraries. And then finally, all of our libraries are validated using next generation sequencing. And again, the cool thing, going back to what I was talking about before, we can actually dream of a library and dream of the design, and then when you actually finally sequence it, it exactly matches. So that's really the power of using algal pools to generate synthetic libraries. So when you think about an antibody library, let's say a VHH or heavy chain only antibody, we can actually make an algal pool that constitutes specific diversity for CDRs 1, 2, and 3, and that diversity could be derived from a natural source. So as I'll show later, it could be derived, for example, from llama-based CDR diversity or human diversity. We can make exact sequences, and then we can shuffle them in the context of a fully human germline framework. So even though we're making a synthetic library, we're shuffling natural, human, or other animal-derived sequences to come up with novel antibodies that bind any target we wish. Another cool thing about using algal pools in this way to generate antibody libraries is I also love to talk about the things that we can take out. We can actually take out CDR sequences that might lead to developability risk or liability, such as dipeptides that might lead to cleavage or isomerization or glycosylation. And then finally, I've also loved to talk about how we can actually also insert specific motifs into our sequences to actually allow us to go after specific types of targets. I won't really talk about that today, but that's actually key and central to many of our GPCR-focused libraries that we have here at Twist. So this slide in a nutshell encompasses the services that we provide for Twist Biopharma. On the left side, you have all of our libraries. As I mentioned, we have 18 libraries, but we're quickly actually innovating and creating more libraries, and we'll actually have over 20 libraries coming up in Q1 of next year. And they're basically broken down based on scaffold type. We have fully human Fab libraries, fully human scFv libraries, as well as a whole host of VHH single domain libraries, which has been really popular lately with a number of our customers. And a number of them really love the fact that we have a breadth of VHH libraries to bear against their particular target. And then, as I said, we also have several different libraries that are focused on difficult to drug classes of targets, from GPCRs to ion channels to carbohydrates. And then on the optimization side, we have a system that we call TAO, which stands for Twist Antibody Optimization, where we can take in any antibody we wish, not just derived from our libraries. We can, first of all, potentially humanize it during this process, and then insert it into our software. And what our software does is it looks at your particular antibody and actually looks at the CDR sequences in particular and finds human equivalents of those CDRs that are within a defined mutational distance that we set. So say, for example, we give it a distance of 3. It'll give us all CDR sequences that are within 3 amino acids of the parent sequence. We make all those oligos and algal pools for each of the different CDR regions of your antibody, and then we can shuffle those natural human CDRs to come up with an optimized human candidate against your particular target. So now I'm going to shift gears and get into why I think deep learning and machine learning is so impactful for our workflow, and why we're trying to add it on to a lot of the discovery campaigns that we've been currently doing using traditional screening. So when you think about kind of the world of sequences that are potentially possible, it's a huge number. Like, you really can't jam all the theoretical diversity of what an antibody could look like into a single library. And we're oftentimes limited to about 10 to the 9th to 10 to the 12th specific sequences, depending on the display platform and how we build the library. But even that's a pretty big number. Through the course of panning using display systems, we typically get it down to on the order of thousands or tens of thousands of sequences. And then we hope at the end of that that we can screen enough to actually pull out all those winners to give us all the binders from our panning campaign. But as I'll show you later, we believe that even though we might screen a lot, and I always believe that if I screen enough, I would get everything. We often miss things that our display system doesn't allow us to actually see through traditional ELISA-based screening. And so if we complement that with the tools I'll talk about today, that'll allow us to really get a lot of those leads that we would have left on the table. So just on a high level, Twist is really trying to incorporate as many technologies to enable us to be successful for our partners. I've talked a lot about our synthetic library offering, and I'll talk to you about how we're using that in conjunction with machine learning. But we've also, last year, actually acquired a mouse immunization company, Veris, to really complement both the synthetic as with the immune-based discovery approaches. And again, a lot of the tools I'm talking about today could also be used for in vivo immune repertoires as well. And then what we find for the machine learning approaches is they will allow us to find, again, those rare hits, as well as potentially in the future, allow us to optimize our candidates as well using the sequences that we generate through next generation sequencing. So overall, for our synthetic workflow, we start with those libraries I talked about before, which again, we typically pull them based on scaffold type. We pan and screen them against a specific target. Typically, we're doing either cell-based or bead-based panning. We'll do about 4 or 5 rounds of panning. And as I'll show later in the presentation, we're sequencing using next-generation sequencing at every round. And so that allows us to see how specific sequences are changing during the course of the panning, from the naive library all the way to the final output from the panning. Once we have all those sequences, again, traditionally we would have used traditional, basic ELISA-based screening. We would sequence them, we would reformat them to the final modality, which in most cases is an IgG, really taking the power of Twist, which is our ability to quickly make DNA as well as quickly make the resulting IgG. We can go directly to that final modality and allow us to find the affinity as well as any function of all those antibodies against a specific target. And we've actually, because we think this is so impactful, we've actually created a group within Twist called, again, high throughput IgG, where you can not only order your specific antibody genes, but also order the resulting antibody as well, either at a 1 mil or 8 mil scale. And then finally, we then take all those antibodies and put them through rigorous affinity ranking, as well as cell-based testing, and sometimes even potentially functional testing to enable us to find our leads. And so, as I mentioned, what we really want to focus on now is by sequencing more in depth, can we find other leads that we would have missed through if we had just done traditional screening? So, again, I'm new to the field, so I'm learning all these terms. But again, machine learning is our ability to use computer systems that can learn and look at the data and find trends and look for how specific sequences, again, are changing through the course of your panning and your screening. And kind of in a simplistic way, these models allow us to kind of narrow down the field of possible sequences that should be or potentially may be hits against your target. The hit rate from a lot of these models is typically not as great as traditional screening. Like, for example, you might get a hit rate of, say, 20 or 30% from your initial models. But the cool thing about them being models is you can always go back, put the data back in, and actually make a better model for predicting potential leads from your data. And so the way I see machine learning models, if you kind of think about it as this diagram on the far right, where you have this ramp kind of going up to the potential lead, the bottom being the naive library where you don't really know exactly what your hits are. Machine learning kind of helps you get further up the ramp towards your specific hit and can, again, narrow down the field of possibilities that actually could be hits against your target. So and we've been using machine learning for a lot of different applications. I'm really going to focus on lead picking today, but these are the ways that we're really planning on utilizing these strategies in the future. On the most diverse end, we've been doing a lot of collaborations with, for example, Deep CDR, now owned by Alloy, to actually help us with generating sequences that can actually be used to generate a library. So can you use a data set like a diverse antibody repertoire to generate a whole series of other antibody sequences that you can then use to build upon and build a library? The second is what I'll spend the most of time talking about today, which is helping us with lead picking from our panning and screen that we do from our libraries. Again, allowing us to find leads that we would have missed through traditional screening. The third area is a really hot area that we're really putting a lot of time and effort into. I can't say we have a technology really to roll out yet, but we're getting there, which is basically allowing us to utilize the sequencing data as well as all the actual binding data and functional data that we generate through all of our leads to actually optimize our candidates further. Can we actually build models that allow us to make specific changes in those sequences to actually, without actually even building a library, to actually do affinity optimization and improve upon their potency in silico? And then finally, I won't show any data today, but we're also working again in conjunction with our in vivo folks at Veris, really trying to use developability data to also generate machine learning models to help us predict developability. One of the cool things about Veris is they've actually generated a lot of this data where they have sequence to developability assay data. And we're actually utilizing a lot of that data to generate machine learning models around developability to allow us to predict whether an antibody might, for example, score very poorly on a Hick-based assay. Okay, so the proof of concept I'll talk about today is a panning that we did with our VHH H-shuffle hyperimmune library. It's a humanized VHH library where we actually have llama-based CDR diversity in CDRs 1 and 2. Framework 2 is still the llama framework so that we can utilize and get solubility of the VHH domain. And then we actually incorporate a very large oligopool over 2.5 million heavy chain CDR3s derived from the naive and memory human B cells into the CDR3 position of that library. So it's got a lot of human content from the framework as well as the human heavy chain CDR3s that are part of it. We then pan and screen this against a proof of concept target, TIGIT. And what we did traditional phage-based screening to find all of our hits that our initial hits through traditional screening, but then we also complemented that by doing next generation sequencing and all the techniques I'll talk about to pull out even more hits. So again, our approach is we'll take the library, do multiple rounds of panning against, in this case, TIGIT. And in this case, we're doing bead-based panning against a recombinant protein target, doing both NGS analysis as well as Sanger analysis on ELISA-based hits. We're formatting those directly to VHH FCs, purifying them on protein A, and then putting them through a whole battery of tests, in particular affinity and expression levels. And again, what we're trying to see is what are we missing through the course of doing NGS analysis and machine learning that we wouldn't have picked up through the traditional ELISA-based approaches. So one technique that we initially do with the NGS data is what we call enrichment analysis. So we look for specific sequences that are enriched through successive rounds of panning. And we have a graphical way of showing this data where we can actually plot the abundance of specific sequences or closely related sequences with, for example, round 1 being on the x-axis and round 2 being on the y-axis. And then we're looking for sequences that actually move up through going from round 1 to round 2. And again, we can look at that throughout the rounds of panning from round 2 to round 3, round 3 to round 4. So again, we're looking for highly abundant sequences that dramatically get enriched through the rounds of panning. And that's one bucket of sequences that we make. And again, typically from this bucket, we'll make at least 100 sequences that are highly enriched. And again, normally you would expect that most of the sequences that you would get through traditional screening would be in this particular bucket, and that is indeed true. A lot of the sequences that we see through traditional screening fall into the enrichment bucket, but there are still some that don't make it through, and so again, it's still good to pick additional clones based on enrichment. The second way is clustering sequences based on features or similarities between the CDR sequences of the various leads. So trying to find sequences that have differences from one another in similar families based on specific features or criteria that we can set around those specific sequences. So that could be clustering sequences based on hydrophobicity of the amino acids, charged nature of those amino acids, And so that allows you from a kind of holistic property level of those sequences, rather than just explicit numbers, to actually find sequences that are coming up through the course of panning based on those specific features. So this is again, would be kind of bucket 2 of sequences that, again, we would probably make about 100 sequences. And then finally, the last area that we want to pull sequences from is from a deep learning model. So here, we can drop in the diversity from the naive library. We can then also bring in the multiple rounds of panning and to see how the sequences are narrowing in on specific sets of sequences through the rounds of panning. And then we can build a model to actually predict which sequences would be hits and which ones would not, based on some of the initial data that we've generated before we actually generate this model. And so in this initial model, it'll allow us to potentially pull out and predict certain sequences that we should make and test. And as I mentioned before, from these initial models, typically we see a hit rate of about 20 to 30%, whereas traditional screening, you might see even upwards of, say, 60 to 70%. So even though the hit rate is lower, this is allowing us, as you'll see in a second, to find sequences that are very rare in the output, but are still predicted to be hits based on the model that we generate. And as I said before, as we talked about before, once we go through an initial round of creating this model, we can go back and bring the new data in to actually create an even better model to predict sequences, again, that we would have missed. So as I mentioned, we took all of those sequences from the traditional screening, the enrichment, clustering, and deep learning. We made over 100 antibodies from each of those different buckets of sequences. And then we put them all through the same series of tests, from expression to, again, looking at sequence analysis, put them through, rigorous developability tests, as well as, in particular, affinity, as well as functional competitive analysis assays as well. Shown here on the right is just some readout on expression, and as you can see, we're getting really high levels of expression from a 1 mil transient transfection, really showing you the power of this library to generate very stable VHHs against particular targets. I really love this slide because it breaks down the relative contribution of these various buckets in terms of traditional screening, enrichment analysis, clustering, and deep learning. And they're all color-coded on the top left based on the algorithm that's used. And on the bottom axis is looking at clonal abundance. So what we see is that, and then on the left, on the y-axis, we're looking at affinity. So what we see is that a lot of times the most abundant clones, either from traditional screening or the enrichment analysis, do show high affinity, but there's a broad range of affinities from that analysis. As we start to go into the enrichment and clustering analysis, we do also pick up a lot of sequences that are less abundant, but also very high affinity. And then finally, when you get to the deep learning models, the neural network models, you see, again, kind of more of a broad range of specific affinities, but again, you can still find very rare clones that have very high affinities. So overall, you can see that if you basically put a threshold of affinity, you can get nice contributions from all of these various buckets of sequences from these 4 different outputs. And again, we get a really rich data set now, whereas before, if we had just done screening, we would have only gotten a slice of the data I'm showing you here. And so shown here is just looking at the sequence analysis. We actually like to compare our sequences using this phylogenetic wheel, where we can look to see how the sequences are related to one another based on primary sequence. We also can layer on a lot of other data on top of that to show you how their affinities, as well as the competitive analysis data I'll show you in a second, as well as their expression levels. So using this graphical way of looking at our data, we can actually home in on specific sequences to go after. Shown on the right, we also ran a competitive ELISA assay looking at competition with TIGIT and its ligand. And again, we find that there's sometimes a really nice correlation, really dependent on epitope, between affinity and competitive analysis in this assay. And it allowed us at the end to find very high affinity as well as very potent antibodies against the TIGIT CD155 interaction. And so as I mentioned, machine learning, you don't have to stop with the first model. We can actually go back, bring back all this data that I just showed you, and we can generate a much better model where it might allow us to go back and find other sequences that we've missed, potentially even allow us to think about ways to vary those sequences to make them even better based on the families of sequences that we're finding from that data. And so, again, I'm not going to go into the fine specific details of the second generation model, but it's really cool to see how we can generate an initial data set, very quickly generate a second generation model, and then again, taking advantage of the power of Twist to make and test antibodies, we can very quickly iterate and find even more hits against our target. And this just shows you how going from model 1, V1 to V2, we can get a much tighter prediction of specific hits from the first generation model to the second generation model. And so again, really shows you this iterative nature of building models and making and testing antibodies as we move along this discovery paradigm. And so in totality, our vision is that we will use all of these various tools to take advantage of all the data that we have. So as I mentioned, we're really pushing and working with our customers to actually make them as successful as they possibly can. As I mentioned, I thought doing lots and lots of screening was the path to doing that, but I'm actually sold now that a lot of the sequencing-based techniques I talked about today is actually really critical to being successful in these campaigns, and it gives us so many more hits against our targets that, again, we would have missed. And that is exactly where we're going in the future. We're going to be kicking off a whole host of ML-based services that are going to be coupled with our discovery services that will enable our partners to come up with hundreds and hundreds of hits to their particular targets. So just to summarize, Twist Biopharma, we're a division of Twist Bioscience. We're really empowered by our ability to make high-quality libraries as well as our ability to make unlimited genes. These machine learning and deep learning algorithms are really allowing us to extract, in totality, all of the hits from our campaigns. I hope you can appreciate from the case study I showed today that we really were able to generate so much more data using these techniques versus traditional screening alone. And again, we're really driving to that Twist Biopharma is going to be your one-stop shop for discovering antibodies derived from mice, from libraries, and again, it'll all be bound together by all these techniques of doing next-generation sequencing and machine learning. Just like to thank the team that did all this work. We really do love VHH domains so much so that we actually hang out with llamas about once a year. So I actually know this guy out in Sonora. He has a llama farm. He brings in 4 llamas every year, and we hang out with them for the day. Really showing you that we appreciate what llamas gave us in terms of their awesome antibodies, and again, that's why we try to model a lot of our synthetic libraries after what the llama creates. I'd like to thank you for your time and attention, and I'd be happy to answer any questions. Sure. Can you hear me okay? I can hear you.
Nice talk. In the phylogenetic wheel, I was trying to visually quickly check where the enriched antibodies are, and they looked pretty evenly scattered, actually. So I guess my question is, how often do you find using these methods to find the otherwise missed hits, that you're really finding new clades or new, sufficiently diverse shots on goal?
Yeah, so we actually use that as part of our selection criteria too. We try to get as much sequence diversification as we possibly can through this process. So that's probably why you saw such a diversification of sequences amongst the highest affinity clones. So that's definitely part of our process.
And maybe just one other follow-up. I mean, in my experience, the highest affinity clones coming out of the initial selection or immunization are not necessarily going to be the winning lead. Sometimes a clone with a different binding mode or a slightly different epitope that starts off lower affinity could end up being your lead. So how do you tend to think about that?
Well, I didn't really talk about it today, but we love to do epitope binning as well on the Cartera. That allows us to really break down the binders in terms of their relative binding sites. Of course, affinity at each of those binding sites is important. So because the binding site then correlates with the function. So you're right. I think within a particular binding site, there will be a correlation with affinity, but there might be another site that's more functional that may not require such a high affinity as another site does. Great. Thank you for your time and attention. If you want any more information, please drop by our booth. And again, we'll have lots of posters as well aligned with a lot of these technologies. So thank you for your time and attention today.